Okay, all right everybody, today we are going to be talking about amino acids. So as you know, amino acids are the monomer units that make up proteins, or you may also hear them called as polypeptides or peptides. So normally biochemists utilize peptides when they're talking about very small proteins, such as, I don't know, maybe 2 to 4 amino acids linked together. Polypeptides may be up to about 100 amino acids linked together, while proteins are much larger, so over 100 amino acids. But sometimes you'll also see people will interchange the terminology as well, but I just kind of wanted to make you aware of the different terminologies that people use, and that they do sometimes refer to specific length ranges. Okay? So, here are your learning objectives for this chapter. Again, you know, after reading the material, listening to the lecture, talking in class, make sure that you can answer these learning objectives to help you prepare for the exam. Alright, so polypeptides, or proteins, are composed of amino acids. So here is our basic amino acid structure. You have your alpha carbon in the center with a carboxylic group, an amino group, a hydrogen, and then some sort of R group or functional group coming off of the alpha carbon. Now this is what changes or varies from amino acid to amino acid. So all amino acids have the same basic backbone structure with varying R groups. Now at physiological pH, we find that because of the pKa of the carboxylic group, it is deprotonated and therefore is negatively charged, and the amino group is actually protonated and thus positively charged at physiological pH. So this actually makes amino acids dipolar, meaning they have both a positive and a negative charge. And another term for that you may see in the textbook is a zitero-ion or zitero-ionic, meaning that they have both the positive and negative charge on the molecule. Okay? Alright. Well, I think I spelled that wrong. So it's actually Z-W-I-T-T-E-R-I-O-N-S. Zitero-ions. Alrighty. So let's erase this guy, because I don't want you all to get confused. Oh, it's taking forever to erase. Okay, let's just continue on. Alright, so when two amino acids, say amino acid 1 and amino acid 2, come together, you have a condensation reaction where water is lost. So we lose this hydrogen and the negatively charged oxygen on the carboxylic group, and we form our peptide bond. Now, in polypeptides, you will end up with an N-terminal, the N-terminus, and this C-terminus at the very last amino acid. So, in proteins, you get linking of amino acids, basically from head to tail, or amino to carbon end, 1, 2, 3, 4, 5, to eventually you have your carboxylic end and your amino end, and then they have the R groups, like so, coming off. Okay? Now, we need to talk about what these R groups are. So basically, your 20 common amino acids can be broken down into 3 categories. The first category is hydrophobic amino acids, or amino acids that have very hydrophobic, non-polar functional groups, such as alanine, isoleucine, and phenylalanine. And if we look at our electron density or our polarity, we can see that, we definitely have a polar region here with the carboxylic and the amino group. However, the R functional group, we can see is very non-polar in all three of these. So, and there's not just three, there's more. So let's go look at our table. So other hydrophobic or non-polar we have is glycine. And what's interesting about glycine is that it just has a proton off of that alpha carbon. Alanine has a methyl group. Valine, leucine, and isoleucine. Okay? We also have methionine, which has a sulfur. Proline is unique in that its functional group comes back and binds with the amino group on the backbone of the amino acid. Phenylalanine, we have an aromatic group. And tryptophan has this imidazole. Is that how it's? I'm really bad at pronouncing. It has an imidazole group. Okay? So again, these would be very, very hydrophobic and nonpolar because of their aromatic rings and the conjugation. Now, we also have hydrophilic, non-charged amino acids. So these are amino acids like serine or glutamine. And again, if we look at our electron density mapping, we can see that we have some hydrophilic or polar residues in the R group as well. So amino acids that fit into this category are serine, where we have the CH2 and then hydroxyl group, threonine, asparagine, glutamine, tyrosine, again the aromatic ring, but we do have an hydroxyl group. So we get a little bit of hydrophobicity because of the aromatic ring, but we also have this hydroxyl group that can hydrogen bond with water. So it's a phenol group. Cysteine, which has a sulfohydryl group. This is the CH2CH group, and we're going to come back to cysteine and talk about why this amino acid is special. And then our third category is our hydrophilic with charged amino acids. So these are amino acids that can lose a proton and gain a formal charge at physiological pH. So those are lysine, with that positively charged amino group at physiological pH. Arginine, again, we have two amino groups, one of which is going to be positively charged at physiological pH. Histidine, again, imidazole. Aspartic acid is going to be negatively charged at physiological pH. And glutamic acid, again, negatively charged at physiological pH. So really, understanding these amino acids and their functional groups can help us to understand how proteins fold. They may fold to allow these very hydrophilic charged groups to be on the outside of the protein, while the very hydrophobic groups may be buried in the inside of the protein. Also, with some of these amino acids that can form charges or interactions with other molecules in the cell, they may actually provide function in doing chemical reactions with other components of the cell, such as other proteins, small molecules, or even nucleic acids. So, for instance, cysteine is really interesting. If you get two cysteines close together, you can oxidize and remove water, in order to form a disulfide bridge, which is a covalent bond between two neighboring cysteines. We see this happening a lot in tertiary and quaternary structure, and we can also see this occurring in certain chemical reactions that we'll find and talk about in metabolism. Now, another important thing to understand about amino acids is amino acids are chiral. So what do we mean by chiral? So for those of you who need a refresher on the chemistry of chirality and stereochemistry, just for the purposes of this class, what you need to understand is chiral centers are carbons that have 4 different substituents. Okay? And what that means is you cannot have a superimposable mirror image of a chiral center. And there's also some discussion in the book talking about how chiral centers can rotate plane polarized light in different directions. So based on how the functional groups are arranged about the chiral center, you can either have an L chiral center or a D chiral center. And that stands for levorotary or dextrorotary. And that just has to tell you which direction that these chiral centers can rotate plane polarized light, or that they, or if you want to think about it, they're the opposite of each other. Okay. So why is that important? Well, like I said, amino acids are chiral. As you can see here, we have the carbon, the carboxylic group, the amino group, the hydrogen, and then your R functional group. However, there's one amino acid that is not chiral, and that is glycine. Why? Well, if you go back and remember glycine's structure, its R group is a hydrogen. Meaning now we have 2 substituents that are the same, so this carbon, the alpha carbon, is no longer chiral. Alright, so just keep that in mind whenever we are talking about amino acids and chirality, that glycine is not chiral. So it is a unique amino acid. Okay? Now, besides that, all other amino acids are chiral, and what's interesting is that all amino acids and proteins are L amino acids. So they have the levorotary designation. In organic synthesis, if you're synthesizing chiral molecules, you're usually going to end up with a mixture of L and D molecules. However, in the cell, the synthesis of these amino acids and of these proteins is so specific that cells will produce only one of the 2 stereocenters, and it seems like cells always produce the L. Now, the book does go into an interesting discussion where certain bacteria will actually make proteins that are D amino acids instead of L amino acids, and that these proteins will be normally found on the outer membrane of the bacterial cells. And what that does is it enables the bacteria cell to survive because the eukaryotic enzymes cannot react with the D amino acids from the bacteria. It won't be able to bind and degrade those enzymes like it could if it was L amino acids. So our enzymes in our body are very specific to the stereochemistry of the substrate, and that's why when our enzymes in our cells produce proteins and amino acids, it's always the L amino acid form and not the D form. Now, this is also important when we talk about medicine. So again, remember when I said in organic synthesis, we usually produce a mixture of stereo-centered compounds. So ibuprofen, for instance, has a chiral center here. And when ibuprofen's produced, it's about a 50% D, 50% L. Now, when you actually get a 50-50, this is called a racemic mixture, meaning it's equal D and L. Not all organic synthesis will give you 50-50, but usually most of them do. In order to produce organic compounds that are 100% either D or L, you have to use chiral synthesis procedures, and usually it's very costly, or you have to be able to purify one out. Now, most of the time, when we're making medicines such as ibuprofen, one of the D or the L, one of the two stereo centers will be biologically active and do what the medicine was designed to do. So with ibuprofen, reduce inflammation and, stop pain. And the other stereo center, such as the L, or I guess in this instance the D, is actually inactive. Okay? And does nothing. So it can have a biologically inactive and then a biologically active component. When this happens, pharmacy companies are happy because then they don't have to worry about purifying out one of the chiral centers over the other, and it makes the medicine much cheaper because now there's not as much work in the synthesis and the purification of the racemic mixture. However, in some instances, such as thalidomide, the biologically, what would be inactive stereoisomer may actually be detrimental and cause damage. For instance, thalidomide is a drug that was given to pregnant women, I believe in the 60s or the 70s. And the active stereoisomer of thalidomide was used as an anti-nausea medicine to help pregnant women cope with morning sickness. However, the other stereoisomer of thalidomide wasn't biologically inactive. It actually caused massive birth defects in the babies of these women who were given thalidomide. So this drug was obviously pulled off the market. I think the manufacturer of thalidomide got sued for quite a lot of money. And what it ended up causing is babies to be born with missing limbs. So they were either missing arms or missing legs altogether. Another interesting aspect to the racemic mixture is a lot of times a pharmaceutical company will come out with a new drug. I believed this happened with Zyrtec. And so when they first come out with a new drug, they patent it. And it will be the racemic mixture. So it will be the active ingredient or the active stereoisomer. And then the other stereoisomer they found to be safe and inactive. And so they will package that as the complete mixture in the pill. When their patent is almost up, the pharmaceutical company will then come in and purify out the inactive stereoisomer and repatent the medicine now as a different medicine, which allows them to extend their patent and reduce competition for the next 10 or so years. Okay. Amino acids can also be modified either with phosphorylation, methylation, hydroxylation, so on and so forth. And what a lot of this does, it can either provide further signaling if a protein is like a neurotransmitter or is on the surface of the plasma membrane. The phosphorylation may be an additional functional group that makes this molecule more unique so that other molecules that are outside of the cell or into the side of the cell can distinguish it from other proteins. And it can also provide more modes of chemical reaction by putting on these extra functional groups that can cause chemical reactions to occur in the cell through that functional group. We'll get into that more when we start talking about metabolism. So a lot of times an enzyme will be inactive until it is phosphorylated. And then upon phosphorylation, the enzyme becomes active and can now drive the reaction forward. Or phosphorylation may turn the enzyme off. So we can control enzyme activity through modifications such as phosphorylation. And we'll talk more about that, but I just wanted you to be aware about amino acid modifications and that it can affect the protein function and structure. Now, amino acids, sort of like DNA, can play other roles than just being monomers for a polypeptide or a protein. Certain amino acids, like histamine, Dopamine are neurotransmitters, and some of them can also be hormones. Again, I don't want you to like commit these structures to memory, but I just wanted you to understand that amino acids can play other roles as just the monomers, much like monomers of DNA, nucleotides can play other roles in the cell as well. Okay, so that's it for today. Make sure you guys go over your pre-activity for Monday so that you come into class prepared. In this activity that we're doing, you're going to be looking at amino acids and using amino acids as buffers. So if you remember, if you go back to your table, you will see that amino acids have pKa's, right? So we already know there's Zitterer ions because we have a pKa for the carboxylic, a pKa for the amino group, and then some of the side chains have pKa's as well. So remember, a weak acid or a weak base that has multiple pKa's are called polyprotic acids, and a lot of amino acids are polyprotic, okay? And so, they have either two or, well, I guess all amino acids are polyprotic. All of them have at least two pKa's, one for the carboxylic, one for the amino group, and then some of them have an additional pKa because of their arc group. Okay, so just kind of keep that in mind as we are preparing for class on Monday, and I hope y'all have a great weekend, and thanks for bearing with me on getting this lecture to you a little bit later than normal. Okay?